D. Insertion of a foreign DNA fragment into a vector. The DNA thus isolated is above or procured from DNA or gene bank is fragmented by using the specific restriction enzyme to develop specific cohesive ends. The cloning vector, e.g. PBR322, is also treated with the same restriction enzyme so that the cohesive ends generated may have complementary residues similar to foreign DNA. The plasmid contains AMPR and tetra marker genes to confer resistance against antibiotics, ampicillin and tetracycline respectively. Some time after cleavage by restriction enzymes and ligation of foreign DNA fragment with the plasmid, a few important genes are destroyed in the resultant product. For example, BAMHI destroys tetra marker genes. The sticky ends can permanently join if T4 DNA ligase, which forms a phosphodiester linkage between free 5' phosphoryl and 3' O group, is added along with ATP at 4 to 100 C for a long incubation time. A number of products are formed from this reaction, which are highly heterogeneous mixture of recombinant DNA molecules together with parental plasmids. For insertion of double-stranded cDNA into a cloning vector, it is necessary to add to both termini single-stranded DNA sequence which should be complementary to a tract of DNA at the termini of linearized vector. In order to get efficient formation of recombinant DNA molecules, addition of sticky ends on both termini is necessary. There are two methods for generation of cohesive ends on the double-stranded cDNA: use of linkers and homopolymer tails. A. Use of restriction enzyme linkers. Linkers are the chemically synthesized double-stranded DNA oligonucleotides containing on it one or more restriction sites for cleavage by restriction enzymes, e.g. EcoR1, HIN3, BAMH1, etc. Linkers are ligated to blunt end DNA by using T4 DNA ligase, figure 6.4. Both the vector and DNA are treated with restriction enzyme to develop sticky ends. The staggered cuts, i.e. sticky ends, are then ligated with T4 DNA ligase with very high efficiency to the termini of the vector, and recombinant plasmid DNA, chimeric DNA, molecules are produced. For details see Chapter 4. See figure 6.4. Insertion of a foreign DNA fragment into a plasmid by using restriction enzyme linkers. B. Use of homopolymer tails. D. A. Jackson and co-workers for the first time in 1972 applied this method to construct a recombinant DNA by inserting phage DNA into SV40 DNA by using DA-DT homopolymers. Using terminal transferase, the synthesis of homopolymer tails of the defined length at both 3' termini of double-stranded DNA and vector can be done. In the presence of precursor DATP, terminal transferase helps to add poly-DA at 3' termini of vector. Likewise, the same enzyme adds poly-T at 3' termini of DNA molecule when precursor TTP is present. The linearized vector having tails is incapable of recircularization, unless ligated to a double-stranded DNA fragment. Vector and DNA tails are annealed. The poly-DA-DT tails are then ligated by T4 DNA ligase, figure 6.5. If poly-DG-DC tails are used, instead of poly-DA-DT tails, a high initiation temperature, up to 37 C, is required for the annealing reaction. The major advantage in using poly-DG-DC tails is that the hybrids are more stable than poly-DA-DT hybrids. It has also been described in Chapter 4. In the first method, the inserted DNA molecule can be retrieved by using restriction enzymes, e.g. EcoRI, on the cleavage sites of linker, whereas in the second method, however, it is very difficult to retrieve the inserted DNA fragment due to the loss of recognition sites when poly-DA-DT tails are added. In the second case, recovery of the inserted DNA fragment is possible only when poly-DG-DC tails are used, because it gives recognition site for restriction enzymes. See figure 6.5. Insertion of a foreign DNA fragment into a plasmid by using homopolymer tail. A. Transfer of recombinant DNA into bacterial cell. Several methods have been developed for introduction of recombinant DNA molecule into host cells. According to types of vectors and host cells, the methods are adopted. Some of the methods of gene transfer into host cells are briefly discussed below. 1. Transformation. Once a mixture of recombinant DNA is obtained, it is allowed to be taken up by the suitable bacterial cells. Originally, the transformation procedure was developed in 1979 by M. Mandel and A. Higa. The strains of E. coli usually do not have restriction systems, hence it degrades foreign DNA. To escape from degradation, exponentially growing cells are pretreated with Ca12 at low temperature, thereafter DNA is mixed up. The event of entering the plasmid containing foreign DNA fragment into a bacterial cell is known as transformation. 2. Transfection. Transfection is the transfer of foreign DNA into cultured host cells mediated through chemicals or viral vectors. The charged chemical substances such as cationic liposomes, calcium phosphate or DEAE dextran are taken and mixed with DNA molecules. The recipient host cells are overlaid by this mixture. Consequently, the foreign DNA is taken up by the host cells. For details, see liposome-mediated gene transfer in preceding section. For the first time, phage was used to transfer the foreign DNA into E. coli cell, therefore it is often termed as transfection, a hybrid of transformation and infection. The efficiency of transformation is not high as it is influenced by bacterial strain and size of foreign DNA. It has been found that the efficiency of this process could generate about 105 transformants per microgram (gg) of cloned circular plasmid, e.g. PBr 322. It has not been possible to achieve efficiencies of over 108 transformants per gram plasmid. If found that the efficiency of this process could generate about 105 transformants per microgram (gg) of cloned circular plasmid, e.g. PBr 322. It has not been possible to achieve efficiencies of over 108 transformants per gram plasmid. If linear DNA is transformed, it is almost completely insufficient in transformation. F. Selection screening of Recombinants. After the introduction of recombinant DNA into suitable host cells, it is essential to identify those cells which have received the recombinant DNA molecules. This process is called screening or selection. Selection of recombinant DNA cells is based on expression or non-expression of certain characters or traits. The vector of foreign DNA present in recombinant cells expresses the characters, while the non-recombinants do not express the traits. Following are some of the methods which are used mainly for selection of recombinants in E. coli. 1. Direct selection of recombinants. The above features is the direct selection of recombinant cells. If the cloned DNA itself codes for resistance to the antibiotic ampicillin, AMPR, the recombinants can be allowed to grow on minimal medium containing ampicillin. Only such recombinants will grow and form colony on medium that contain AMPR gene on its plasmid vector. Following this procedure, you cannot say whether the recombinants growing on such medium contain relegated plasmid vector or contain recombinant plasmid plus foreign DNA fragment, because AMP gene is present in both the recombinants. See figure 6.6. Use of antibiotics in selection of recombinant. 2. Insertional selection inactivation method. This is more efficient method than the direct selection. In this approach, one of the genetic traits is disrupted by inserting foreign DNA. Antibiotic resistance genes acts as a good insertion inactivation system. As discussed in earlier section, that plasmid PBr 322 contains two antibiotic resistance genes, one for ampicillin AMPR gene and the other for tetracycline tetra gene. If the target DNA is inserted into tetra gene using BAV1, the property of resistance to tetracycline will be lost. Such recombinants would be test sensitive. When such recombinants containing target DNA in tetra gene are grown onto medium containing tetracycline, they will not grow because their tetra gene has been inactivated. But they are resistant to ampicillin because AMPR gene is functional. On the other hand, the self-ligated recombinants will show resistance to tetracycline and ampicillin. Therefore, they will grow on medium containing both the antibiotics. To ascertain the presence or absence of tetra gene in the inserted DNA fragment in plasmid, a replica plating is done from the master plate. Bacterial colonies of master plate are gently pressed with sterile velvet so that a few cells of each colony may adhere on it, which is then pressed on other plate containing the nutrient medium amended with tetracycline. Plates are incubated for the growth of bacterial colonies. The appearance of colonies is compared with the master plate, and those colonies that fail to grow on replica plate figure 6.6a can be said to have a plasmid which had inserted DNA in the tetra gene of plasmid and had destroyed tetra gene. Hence, tetra gene becomes non-functional. 3. Blue-white selection method. This method is used for screening of recombinant plasmid. In this method, a reporter gene Lux is inserted in the vector. The Lux encodes the enzyme sus-galactosidase powerful, contains several recognition sites for restriction enzymes. Beta-galactosidase breaks a synthetic substrate, X-gal-5-bromo-4-chloroindolol Sus-D-galactopyranoside, into an insoluble blue colored product. If a foreign gene is inserted into Lux, this gene will be inactivated. Therefore, no blue color will develop, because Sus-galactosidase is not synthesized due to inactivation of Lux. Therefore, the host cells containing rDNA will form white-colored colonies on the medium containing X-gal, whereas the other cells containing non-recombinant DNA will develop the blue-colored colonies. On the basis of colony color, the recombinants can be selected from the agar plates. Figure 6.7. Figure 6.7. A plasmid vector designed for blue-white screening. A. Colonies produced by blue-white screening. B. 4. Colony hybridization, nucleic acid hybridization technique. The transformed cells grow on nutrient medium, but it is likely that a few cells may have specific DNA as desired among several thousand cells. How is it possible to pick up those cells which have the desired DNA? To make it easy, the colony hybridization technique has been developed in 1975 by M. Christian and S. I. Logness. It has suitable use with plastoids. An analogous method, Plaque hybridization technique, has been given for use with phages. DNA probes. The colony hybridization technique is based on the availability of a radiolabeled DNA probe. A DNA probe is the radio-labeled 3,2-PO4 small fragment of DNA molecule 20 to 40 BP, which is complementary at least to one part of desired DNA. Generally, probe DNA is labeled with 32-PO4 or sometimes with 125I. The 32P liberates Sus particles, while 125I emits gamma rays. In addition to using radioactive isotopic elements, isotopic sulfur or fluorescent molecules may also be used. Therefore, a DNA probe recognizes A, T, G and C nucleotides and combines with the complementary sequences of the target DNA. A and T of the probe combined with T and A of 9T get DNA and vice versa. Similarly, G and C of probe combine with C and G of target DNA and vice versa. The use of DNA probe in searching the functional target DNA is given in nucleic acid hybridization technique. The small-sized DNA probe easily catches the target DNA for hybridization than the long-sized probe. Besides, a high rate of hybridization is achieved on maintaining higher concentration of 32 than the target DNA molecules. Moreover, the DNA probe is very specific to its target DNA. This specificity is called stringency. The probe may be partially pure mRNA, a chemically synthesized oligonucleotide, or a related gene which identifies the corresponding recombinant DNA. DNA probes have commercial significance. They diagnose specific DNA sequences, genes, and are used in the diagnosis of diseases and microbial tests. See the figure. Figure 6.9. Colony hybridization technique. B. Colony or plaque hybridization technique. Colony hybridization technique was developed by M. Grinstein and D. S. Hognes in 1975. In this technique, master plates are prepared as described earlier. Replica plating of colonies is done onto a nitrocellulose filter disc Fig. 6.9a, which is then placed on the surface of a gelled nutrient medium, and both master plate and disc are incubated to develop colonies. Cells growing on nitrocellulose filter disc are nourished through the diffusion of nutrients from gelled nutrient medium. Figure 6.6b. The filter disc is removed and put on blotting paper soaked with 0.5 NaO solution. The alkali diffuses into nitrocellulose, lyses bacterial cells and denatures their DNA. Thereafter, the filter disc is neutralized by tris-hydroxymethyl aminomethane HCl buffer by keeping high salt concentrations. Then the filter is dipped in proteinase K solution that removes proteins from the filter. This results in binding the DNA with nitrocellulose disc in the same pattern as the bacterial colonies. To fix the cDNA properly, the filter disc is baked at 80°C. Figure 6.9c. Then it is incubated with a solution containing radioactive chemical labeled probe P32 DNA at suitable conditions. The probe will hybridize any bound DNA which contains sequences complementary to the probe. Figure 6.9d. By thorough washing and hybridized, unbound probes are removed from the hybridized probe colonies containing sequences complementary to probe, and is identified by autoradiography of the nitrocellulose filter disc. Figure 6.8. Colonies which develop positive X-ray image. Figure 6.9g are compared with master plate and picked up, and multiplied on the nutrient medium. See Plaque lifting method. In 1977, W. D. Benton and R. W. Davis devised this technique for screening of bacteriophages from plaque-forming units. The recombinant phage particles are isolated and used to construct DNA library. In this method, lawn of E. coli is prepared on agar medium which is allowed to get infected by recombinant phage particles. They infect E. coli, multiply inside cells and lyse them, forming plaques. As described in Southern blotting technique, replica plates are prepared from the master plate containing plaques by using nitrocellulose filter paper. This filter is treated with alkali solution so that the phage DNA may be denatured. Then filter is baked at 800 C to fix denatured phage DNA and put in a solution containing 32 probe. After some times the probe hybridizes the desired DNA if complementary bases are present. The filter paper is washed with standard saline citrate solution to remove an hybridized probe and passed through autoradiography. Dark spots are formed where probes have hybridized the desired DNA. Then the recombinant phage particles can be isolated from the plaques. 5. Selection of recombinants transgenic tissues. For detailed discussion see Chapter 13. 6. In vitro translation. In vitro translation is now used as a method to confirm the identification of recombinant clones. S. Nagata and co-workers in 1980 have used primary translation screen by isolating the interferon gene from total leukocyte poly A plus RNA. Translation of poly A plus RNA is done when it is microinjected into oocytes from the toad, Xenopus laevis, with the result of secretion of interferon into culture medium. Interferon is detected by its antiviral activity. It is supposed that the number of interferon mRNA may be between 103 and 104, therefore, about 104 transformants must be repeated to get at least single clone. Thereafter, DNA is immobilized on nitrocellulose or diazobenzyloxymethyl cellulose paper to isolate complementary mRNA by nucleic acid hybridization as described earlier. The RNA can be eluted from the cDNA RNA hybrid and microinjected into oocytes and the polypeptides as the translation products are detected by immunoprecipitation techniques. It confirms in vitro translation of foreign gene. See figure. Figure 6.10. Steps of immunological test. 7. Immunological tests. The immunological techniques are the final test analogous to colony hybridization technique as described earlier. It is an alternative screening procedure which relies on expression and generally applicable approach to identify a clone synthesizing a particular polypeptide. This is potentially a very powerful method since the only absolute requirement is that the required mRNA encodes a protein for which a suitable antibody is available. 2 in situ techniques given by S. Broom and W. A. Gilbert 1978 and Ehrlich et al. 1978 are in current use. Instead of DNA molecules, antibodies, immunoglobulins, are used to identify colonies or plaques developed on master plates that synthesize antigens encoded by the foreign DNA present in plasmids of the bacterial clones. For this purpose, a vector, known as expression vector, is designed where the foreign DNA is transcribed and translated within the bacterial cell. The medium containing specific antiserum may help in detection of viable immunoprecipitate precipitin around the colonies or plaques. The method follows: 1. The replica plating of bacterial colonies of master plate on nutrient agar. 2. Lysis of cells after their growth by exposure to chloroform vapor, or treatment with high temperature. 3. Making gentle contact of a solid support, for example, a cellulose filter containing immobilized antibody to solid support with the lysed cells within the colonies to allow absorption of antigen to antibody, and 4. Detection of antigen-antibody complex by incubating the cellulose filter with a radio-labeled second antibody. The antibodies that do not react are washed off in position of the antigen-antibody complex as determined by passing the filter through X-ray. It gives the signal of those bacterial cells which synthesize antigen on the master plate. Figure 6.10. See the figure. Figure 6.11. Detection of specific antigen synthesized by a bacterium. S. Broom and W. Gilbert in 1978 purified immunoglobulin G Ig and bound it to an agar plate so that the antigens released through in situ lysis of bacterial colonies can bound to the fixed antibody. Figure 6.11. The antigen is then detected by using the same Ig preparation which 4.6 as radioactively labeled. This recognizes determinants on the bound antigen. Ehrlich et al., 1978, used F-Ab-H fragments derived by digestion of pepsin of the immunoglobulin, bound to either polyvinyl cellulose or diazobenzyl oxymethyl cellulose (DBMC) paper. This is incubated first with the antigen and then with undigested antiserum, FC portion of which will bind radiolabeled Staphylococcus aureus, a protein.